This video is intended for Anatomy and Physiology 2 students, and it's going to summarize a number of videos I have on white blood cells. This is part of the blood chapter, which is significant. I'll tell you why in just a second. So in the bone marrow, there are these stem cells, and they can be stimulated with a number of different hormones, while erythropoietin will cause the differentiation of these cells to produce red blood cells. There are a number of locally acting hormones known as cytokines, which will then cause these stem cells in the bone marrow then to produce a number of different kinds of white blood cell. And so there are different kinds, as we'll go through in a second. When we spin blood down, the blood separates into three layers. The cell-free plasma is the top layer. The hematocrit with the red blood cells is the bottom layer. And in between is the buffy coat, which has white blood cells and platelets. So you spin blood down and it creates plasma, the buffy coat with white blood cells or leukocytes and platelets. Notice that the buffy coat was fairly thin compared to the hematocrit in the red blood cells. And so then one could, not be overly impressed by the number of white blood cells. In a previous video, you had seen there's red blood cells everywhere, and then here's maybe one isolated white blood cell. Here's another isolated white blood cell. The white blood cells were not that impressed by them in the blood. And in AMP2, the blood chapter really only superficially mentions the white blood cells. Why is that? Well, the answer is because they're not in the blood. So if one were to look at blood, you'd see an isolated white blood cell here or there. But if you were to then look under the microscope at lymphatic structures, just as the thymus, the white pulp of the spleen, a lymph node, a tonsil, a Peyer's patch, the appendix, you'd see images like these where all of those cells were white blood cells. And so there simply aren't that many white blood cells in the blood. Most of them are instead in the lymphatic system. And so that's why in this chapter, we really don't cover them to any great length. Instead, they are covered in the lymphatic system chapter. So this is kind of a very superficial view. And if you're disappointed because you say, I want to learn more about antibodies, et cetera, you should. But white blood cells are primarily found in lymphatic tissues, and that is where they are studied. So here we see blood skipping along, and not to be insulting, but red blood cells are stupid. They don't have a nucleus, so where are they going? They don't know. They're going where they're pushed. Blood pressure is taking them along, and they will simply follow. White blood cells are very, very different. And there are two things which are visible here. First off, white blood cells are looking for signals. So for example, if this tissue is inflamed, inflamed tissues produce signals. So here are signals showing that there is a problem. There's an infection, there's tissue damage, there's inflammation. And now these white blood cells can then squeeze out in between the endothelial cells which line the blood vessel. So these white blood cells are demonstrating two properties that red blood cells lack. They have both chemotaxis, they can move towards certain chemicals, such as inflammatory signals or signs of infection, and then they can also emigrate from the blood vessel towards peripheral tissues, which is why the overwhelming majority of white blood cells are simply not in blood. They have left to go to the peripheral tissues. Now, once again, in just this quick summary and in preparation for what you would probably do in lab, we notice that there are different kinds of white blood cells. And the reason for this is quite simple. These are, to a large extent, protecting your body. And if you think of our society, who protects us? Well, clearly there's the Army, the Navy, the Air Force, the Marines. There's the Coast Guard. There's the ATF. There's the CIA. There's the FBI. There are firefighters. There are state police. There are local police. There are local security guards. It is a very long list. Why? Well, the answer is because there's different kinds of threats. What the US Navy protects us from and what the fire department protects us from, or what, say, campus security protects us from, are three very different kinds of threats. And then each have specialized to be able to protect against a specific type of threat. And in the same way, if you ask, as we will in the lymphatic system chapter, what threats face the body, the answer is there's different kinds. There's bacteria, there are viruses, there are eukaryotic protists, there are parasitic worms, there are ectoparasitic insects and arthropods, there are cancer cells. So just as there are a lot of different threats of society, therefore there's different aspects of the society protecting us, so too are there different kinds of white blood cell, each specializing in different kinds of threats. When we look at the white blood cells, one of the first things that's done is to stain them using the right stain. And with this stain, the nuclei stain purple. And then 3 of the 5 major classes of white blood cells then have specialized lysosomes, which we'll call granules, which then also take up stain. So there are 3 which are called granulocytes, because when you look in their cytoplasm, you see these granules, these lysosomes which have taken up stain, while then in 2 of these you don't see granules, so we call them agranulocytes. So the first thing that we tend to do is stain them and distinguish in between these the 3 granulocytes and then the 2 agranulocytes. So here's another view of that. Once again, in the following chapter, these can be covered in greater detail. But first, to go through the granulocytes. Once again, there are 3 of them, and we'll distinguish between the neutrophils, the eosinophils, and the basophils. These are the granulocytes, compared to the agranulocytes. Here is a lymphocyte, and there is a monocyte. Now, just in fairness, the monocytes, that's the term we tend to use in this chapter, but once they leave the blood, then they become larger, and we then talk about them as either macrophages or dendritic cells. Once again, that's part of another chapter. So neutrophils, in a lab, you might have to identify these. And how would you identify them? Well, one, their nucleus then splits into a number of separate lobes, 2 to 6. So if you see 4, 5, 6 lobes of the nucleus, you can say, aha, that is a neutrophil. That is the easiest way to distinguish between them. As we'll cover later, they're actually preparing to use their DNA as a trap. So when there's an infection, these cells actually kill themselves. They burst, their DNA goes all over the place, but it traps the microbes and helps to contribute to pus. And so this, condensation of the nucleus and the lobes is a preparation for that. They are granulocytes because here we can see that there are these very fine lilac granules, which take up a lilac stain in the right stain. So that's how you would identify a neutrophil. Had multiple lobes in the nucleus, sometimes called a polymorphonuclear leukocyte, and then fine lilac granules. The second type of granulocyte is the eosinophil. In the right stain, there is a red stain known as eosin. And eosinophil literally means the lover of red stain. And so there are white blood cells which take up this red stain in large granules, larger than what we saw in the neutrophils. And their nucleus tends to have 2 or 3 lobes. As we'll cover later, they specialize in parasites such as worms. And so they would be very effective against those type of parasites as compared to neutrophils, which specialize in bacteria and things which are bound to antibodies. The final kind of granulocyte is a basophil. Basophils have so many granules which take up a dark purple stain that it actually obscures the nucleus. So the nucleus kind of has, two lobes or an S-shaped, but you wouldn't get to see it fully in most cases, once again, because there are so many of these granules which cover the nucleus. These granules, they have inflammatory signals, and so basophils and their cousins, the mast cells and connective tissue, they primarily function in inflammation. The largest of the white blood cells in the blood is the monocyte. They have a kidney-shaped nucleus, and if you're looking at a microscope slide, the red blood cells are all around, and so the red blood cells can be used for a size comparison. Monocytes can be 2 to 2.5 times the size of a red blood cell. They are agranulocytes because they lack granules. And then in comparison, the lymphocytes are the smallest of the white blood cells in circulation. They have very little cytoplasm. And so they are agranulocytes because they lack granules, and they are primarily observed with this big central nucleus and very little cytoplasm aside. The lymphocytes are extremely complex. These are the cells which produce antibodies, which are capable of recognizing cancer cells, as will be covered later. The monocytes, they have lots of roles, but because of their size, they're very good at ingesting large things like yeast cells, or even old red blood cells or neutrophils, which have eaten bacteria. So if neutrophils fill themselves with bacteria, the monocytes can actually then ingest the neutrophils. So in general, as we ask what these white blood cells are doing, there are lots of things. Certainly certain topics like recognizing cancer and the production of antibodies. This is complex and will be covered later. But to a large degree, many of them perform phagocytosis. They are capable of recognizing a microbe. They ingest it as their membrane pinches inward. And then those granules, which you saw, can contain then the digestive enzymes, which rip the microbe apart and can destroy it. White blood cells also promote inflammation. They can induce fever. So they have a number of different roles in the immune system. Because each class of white blood cell has a type of threat that it specializes in, then there is what is known as a differential white blood cell count. Under normal conditions, the neutrophils are the most common white blood cell in circulation. Lymphocytes are second, monocytes third, and eosinophils and basophils, they tend to be the rarest. And so neutrophil counts, could be 50 to 70% of the circulating blood. The lymphocytes, say 20 to 40%, monocytes 10 to 15%, eosinophils maybe 1 or 2%, basophils less than that. So if this was the distribution of white blood cells in a patient, that is normal. But since neutrophils are effective against bacteria, if one were to notice an elevated neutrophil count beyond this concentration, then one would suspect a bacterial infection. If there were, say, parasitic worms, then eosinophil counts would be elevated, since that is what they specialize. When we spin blood down, not only do we get these 3 layers, but then these layers typically represent a certain percentage of the overall blood. And so if you were to do this manually, you could take a drop of patient's blood, spin it in a glass capillary tube, and then hold it up to a chart where the blood goes from 0 to 100, and then ask, what percentage of the whole blood is the hematocrit. And so some individuals as you might cover more in a lab. They have a higher than normal hematocrit. And in general, men have thicker blood than women. Androgens promote more erythropoietin production. But there are also then other conditions like hypoxia, COPD, or the use of blood doping or erythropoietin and anabolic steroids, which would increase hematocrit. Hematocrit could be then lower than average in women, and in patients, have undergone hemorrhage, anemia, maybe they don't have enough iron in their diet to make hemoglobin, or kidney disease, given that kidneys are where erythropoietin is produced. In the same way, when you spin down the blood and you look at the buffy coat can be thicker than one would expect. And so while a differential white blood cell count is when you count the neutrophil percentage versus the eosinophil percentage, and you can say, aha, the neutrophil count is higher than expected. This might be a bacterial infection. You can also then do a total white blood cell count, where you're just asking in general, how many white blood cells are there in general, and a total white blood cell count would be elevated if, say, there was an infection or leukemia. And so, for example, one of the things that would, if someone had an elevated total white blood cell count, their buffy coat's super thick, they might have been recovering from the Epstein-Barr virus and have had infectious mononucleosis. And so when one looks, say, under the microscope at their blood cells, you would see far more white blood cells than is normal. Now, you'd also see abnormal shapes. These monocytes look sticky. They seem to be adhering to neighboring cells more so than others. But, once again, this is a video which summarizes others. This video goes into mononucleosis in a bit more detail. The point would simply be that you would see more white blood cells following an infection than normal. And then there is also then the possibility that leukemia would then result in an uncontrolled production of white blood cells. So if the bone marrow is where white blood cells differentiate, then there can be genetic mutations which cause these white blood cells to be produced to an elevated level. Now, in the study of cancer, we realized that there are lots of genes involved in cell cycle regulation. So there are lots of then different kinds of leukemia, as you can gather here. So all of these individuals would have too many white blood cells. There would be an uncontrolled production of white blood cells. But as you can see just under the microscope, they're not all the same. And so there might be some which are producing more of the monocyte class types of cells, others which are producing more lymphocyte classes of cells. And so while leukemias share the feature of having an uncontrolled production of white blood cells, there are different classes. If one were to then look at the bone marrow of a leukemia patient, you could see that clearly then here are those stem cells, but far too many are involved in the production of the white blood cells. So there's clearly uncontrolled cell growth here. And so white blood cells are not primarily found in the blood. 98% of them are out of the blood and primarily have located to parts of the lymphatic system. That's why they will be thoroughly addressed in that chapter. But here in the blood, they wanted to give just a quick overview of white blood cells.